what I thought I'd do today is it's not very advanced, but it's about how to make figures using some scripts that I wrote for ImageJ or Fiji, and you can get them via the update site. So I've got Fiji open here, and I just ran the updater, which you can get to from the menu at the top, and it's telling me my ImageJ is up to date. But what you can do is add update sites. I'm sure you guys know this, but there's a whole bunch of them here, and these are third-party sort of sets of code that people have written, and mine is here. So if you just check this box and then you close this window, you'll see a list of scripts and things here, and then you can just click apply changes and it will install the stuff that I'm going to show you today. So I've already done that. In fact, that's how I do the update sites from this computer. So so all my stuff is here, and when you install that update site, you'll get this little menu here called Lab Code, and there's a whole bunch of goodies here, but I'm mainly going to be showing you about Figure Maker today. So it's this menu here, but there's some other utilities and things here if you're interested. There's full details of how this works on my GitHub page where I'm developing ImageJ tools and things. So if anything's not clear, you can go and have a look there. But I think actually this video should be useful to show people how to do this kind of the right way or whatever. So the rationale behind this Figure Maker thing was to standardize the way that we make the figures in my lab. So I was a little bit there are a lot of ways of making montages and figures in Fiji itself that have been written by other people, but none of them, in my opinion, look very good, and they weren't sort of the gold standard for cell biology papers. So this is why I wrote my own. So what I'm showing here is a figure. Actually, the final figure looks a bit different to this, but it's just an example. And what we're going to do today is to generate one of these montages that you see here. And so this is a typical sort of microscopy experiment that's been done. And just to explain, the way we have our figures is we would make them in something like Illustrator, but we would put this in as an entire object. This is one object here, and this is one object here. So we would make two files here for the images and just place them, embed them directly in Illustrator. So the point here is we make one flat image per panel. And that's important because in Illustrator you could put these in as individual things, and you could even try and make your own Roi Zooms in Illustrator. But the problem with that is that it can quickly get out of hand and you can start to make mistakes. And there's nothing more embarrassing than having a figure that's wrong, if you've published it. So the aim of this is to get it reproducible and minimize the number of mistakes that can occur, because it is quite complicated putting these things together. Then what we would do in Illustrator is do the labeling. So none of that is done in Fiji because you get higher quality in Illustrator. We do the big labels on the top, and these would be made in another program like R or Igor or something. We would drop these in as a PDF. Everything is kind of scaled so that we're doing minimal messing around in Illustrator, and that's the way we make the figures in the lab. And the other thing to say is I'm kind of particular about the way we have these figures. So so the separate channels are shown as grayscale, and the reason for that is that we don't show the green channel as black through green, and we don't show the red channel as black through red. That's because your eye doesn't see black through green the same as it sees black through red, regardless of whether you're colorblind or whatever. It's just your eye cannot tell the intensity of two different colors in the same way. And so the best thing to do is to show them as grayscale. So that's what we do here. So we've got 1, 2, 3 grayscale channels for GFP, in this case epsin2, and this protein here. And then we can show them as a merge on the right. So this is the way that our figures are put together. So we typically have the merge on the right. We have separate grayscale channels shown. So I'm going to show you how to do that today. And we could have perhaps other merges if we've got, if we want to do other comparisons. The other thing to say is that we have this grout between the different squares here. So if you use some of the other montage makers, they don't put in a grout, and that looks ugly in my opinion. And there's also a rationale to this, that the grout here is a little bit narrower than the grout here. That's because these are from one protein and this is from a different protein. So it helps you to just see quickly that this is one experimental condition here and this is another, and these are related and these are not. So those are the sorts of things that we can standardize in this figure maker. Just a note about color blindness. So in my lab, we use red, green, blue merges, and we get around the color blindness issue by showing these separate channels. So if you're colorblind and you can't see red as a separate color, then you can just look at the separated channels. But for people who can see all three colors, this merge, particularly when we've got three channels, then green and red make yellow, red and blue make magenta, and blue and green make a kind of a cyan color, and then all three channels together make white. So that's the reason we do it in red, green, blue. We can look at where we've got these complex overlays of three different channels, and we can see them the way they come together with different colors. Okay, so basically that's what the finished product looks like. So how do we get there? So what I've done is I've prepared here some images already that I'll use today. And the point here is that we would typically have an image that is much bigger than this, but we would crop it down before doing the montage making. So in this folder here, I've got pre-cropped images from the microscope. So we crop them square. If I just go back to that original, this final product, you'll see that each of these things here is square, and that's kind of important for the layout. It's good for the aesthetics, but the way that this is dealt with in the code is that if you click this button here, you can click this thing here which says click square ROI. So this will appear when you've installed the update site, and you get this little icon here. Now if you double click that, you can adjust the size of this. This is in pixels. So if we wanted to make an 800 square image, we can do that and then just click on the image in the center and place the square where we like, and then we can either duplicate or crop and save that as a new TIFF to get our square image that we'll use for assembling the figure. So that's what you need to do first of all before running any of the routines. Okay, so the other thing to say is that there's a scaling on this image which came from the microscope originally. So this is 45.48 microns in this direction and the same in the other direction, because the image is 1,000 pixels. We can calculate the pixel size, and that's important, as I'll explain in a moment. Right, so we've got our images, and you can see we've got 4 channels here. We actually have, they're in wavelength order. So we have DNA, there's a green channel here, a red channel, and then there's a far red channel. So we're going to make a montage, and I'm just going to show you that you don't have to use all the channels, and you can combine them in whatever way you want. So I'm going to do this first of all just for one image. So if I go here, click lab code, figure maker, what I'm going to do is make montage. If I have an image open, it will tell me that that's not allowed and ask me to close it, but I don't have any images open at the moment. So what it does is it asks which files do I want to make the montage from. So I'm going to use that one that I just showed you. And it then asks how many grayscale panels do we want. So these are the separate channels that are going to be on the montage towards the left-hand side. So in our final version, we had 3, so let's do that. But you could have up to 4 because there's 4 channels. And it asks how many merge panels you want. We'll just do 1, but you could do a second one if you wanted to compare a couple of different channels. We'll just leave this checkbox unchecked at the moment. I'll explain that at the end. And you just click OK. And then what it does is it gives you the choice, because obviously those grayscale panels, you could put them in whatever order you wanted. And so typically what we do is put them in green, red, blue order and then have the merge on the right. So if you remember, channel 1 was the DNA, so we're not going to show that. What we'll do is we'll put the green here, the red here, and the far red here. And then we can select how we do the merge. So let's put the green in here. So that's 2. This one's 3, and this one's 4. This is slightly difficult to remember because the green, red, and blue are in a slightly different order than here. But anyway, now if I click OK, we'll get our montage. Just to explain here that the grout size refers to that gap between the different squares of the montage, and the DPI is 300, and this is set so that it can be used directly in Illustrator. So 300 dots per inch is the sort of standard, well, it's actually the low end standard for reproduction for journals, so that's why we have this 300 DPI. You can include a scale bar at this point. We won't do that at the moment. I'll do it at the end when we compile the montages together. But the important thing to note here is that the code knows that the scaling of the image. So it tells you here 0.045. So that's correct, because you can see here this scaling for 1000 pixels is 45.48 microns. So this scaling is correct. Now what happens is, once we've saved, we change the scaling to 300 dots per inch, and then the scaling, if we did this again, would read this number here. So you just have to make sure that if it's telling you this, you should just double check what the pixel size is. You should just know what your pixel size is anyway, and if you're going to add a scale bar, just make sure it's right. And the software does its best, but if it's been altered, then obviously the scale bar will be wrong. Okay, so if I click OK, we get our montage, and that's, you know, it's a TIFF, it's at 300 DPI, and it's in RGB color space. So it, you know, this is good to go and put into Illustrator if we just wanted one montage. But what we want to do is we want to make several montages. So it gets saved, by the way, back where the original file was. What we can do instead is, if we've got a lot of these to do, this can be a bit annoying to do them one by one. So there's make montage directory here. So what I will do is I will just delete that one that's already montaged, and we've just got our 3 files that we want to work with. And so we'll tell it these are the ones we want to make montages with, and then it wants to know where to save them. So we can just save them back in the original directory. So we click open there, and then we get our choices again. Now this time the code hasn't read in the file first, so it doesn't know how many channels there are. These files could be anything. So you need to know this about your image before you do the directory version. But we've just done that, so we know that we want to have 3 grayscale panels. We just want 1 merge. Again, we'll just leave this unchecked. This time it doesn't know what these images are called, so you just get channel 1, etc. So we'll do 2, 3, 4, and then we'll do 2, 3, and 4, and we'll get exactly what we got last time, but this time we'll get all 3 versions. So just leave that. Okay, so what you can see now in this folder is we have all 3 files with the montages made. Okay, so that's good. Now we want to compile them into a full image. So what we do is we open all 3 of them now, and we select here Figure Maker Montage Compiler. Now what we're going to do is stack these from top to bottom. And so we can just, it tells us which ones are which, so we can just select whatever order we like. And now the grout is reading 16, so if you remember it was 8 pixels grout going across, and so it's double that going downwards. This gives that nice spacing that I mentioned at the start. Just to say that, you know, that spacing really does depend on the size of that square that you've used. So if you have a 200 by 200 pixel square and you have 8 pixel grout, it's going to look thicker than if you have a 1000 pixel square thing and an 8 pixel grout. So it's all relative, and sometimes you need to play a little bit with this, but what we tend to use is 2 times the grout to give that nice aesthetic look. This time we're going to include a scale bar, so we check this box, and you remember what I said about the pixel scaling. Now this is messed up, and we know that that isn't the right pixel scaling, so we have to manually enter that. You can put whatever size scale bar you like. We tend to use 10 microns for a view of a cell. And we just click OK. And the montage is made. So it's in the order that we said, and the scale bar is in the corner. And this is ready to just go straight into Illustrator. The other thing to say about this is we get a little text file when this is saved, and it tells us which montages are in which row, how big the scale bar was, what the pixel size was, and how big the grout was. I added this recently because having had to go back and remake a whole bunch of figures for a paper, I realised I didn't quite remember all the details that I'd used when I put everything together. So this is a way of enabling us to reproduce whatever montages we make. Okay. So that's basically how to use the Montage Maker. So I'm just going to show you a couple of variations now of other things that you can do with it. So one thing we can do is we can make inverted montages. So this came up in a recent paper of ours where the signals were quite dim and the grayscale was mainly black and there were very few white spots, and we wanted to invert those grayscale channels. So I'm just going to show you how to do that. So in here we can say invert montage, or we can do invert montage auto. So what I'll do is I'll show you, I think I'll just do auto because it's quicker. So I load all those montages that I want to change, and now I just say invert montage auto. And now what we get is different versions of those montages that have been inverted. So the black to white is changed from white to black, and we now have a border on the inside of each of those frames because because the image is mainly white, and with a white grout it looks a bit weird if you don't have a border. So there's the border there. From those inverted montages, you can then compile them into a figure as we did before. In fact, I'll just quickly do that so I can show you how it works. So again, we just do Montage Compiler, pick the same ones as we did before. We'll leave the scale bar this time, and we'll just do it. And we get a new montage compiled, and a new text file telling us what's what. Okay, so that's how to just quickly change everything and put the figure back together. If you do the version, the other version, I'll just quickly show you this. If we do invert montage, you do actually have a bit more choice than on the auto. The auto version just assumes that you have one merge on the right, and it'll just invert the other 3 or however many there are there, and it'll do the black border. Whereas you might not want to invert everything. For example, you might want to leave this one. You might not want the black border. So there's a few options there. The last one for the merge is unchecked as default because you don't want to invert this probably. You just want to invert the grayscale panels. Anyway, so that's how to do that. The other variation that you might want to do, and was shown on that original image, is we have these Roi Zooms shown in the figure. So I'm just going to show you how to do that quickly. So if I go back to my montage here, actually that's a bad one to do. Let's take this one. Okay, so we typically want to add these Roi Zooms, so we would have a little box around a feature that we want to expand and put into the corner of the image. So to do that, you open the image that you want to work with, and you click Roi Zoom, and then it asks you what corner do you want the zoom to be shown in. So it's either left top, left bottom, right bottom. So here we've got a gap in the left bottom, so we'll stick that there and it will go here. Now the sizes depend really on the size of the image. So 50 pixels of 1000 is actually really small, and a 2x expansion is only going to be 100 pixels. So that'll be like 1/10 of the width there. So let's make that a bit bigger. 75, expansion of 4. We'll keep the border for boxes being 4 pixels. I'll just leave this white border checked for the moment, and I'll show you what that's about in a moment. And then you have some control over where you put these boxes and zooms. So the default is to put them in all panels, but maybe you don't want to do that. So for example, if you've got DNA in one channel, you might not want to have a box showing with nothing there or whatever if you've selected a region outside the nucleus. So maybe you want to uncheck one of these boxes, but otherwise you can leave them all checked. And then it asks you to place the center of the box. So let's say we're interested in this spot here. We can place our multi-point tool there. You can move it around if you're not happy, but you just get one shot at doing this. So it's like that. We click OK, and then the code puts a box around our region of the size that we said, so that's 75 pixels, and it does the expansion we said and puts it in the corner that we specified. And also there's a text file made to tell us what happened, so what the expansion was, what the size was, and what corner. And again, that's to help you put these things back together. Now you can combine those, as long as these montages are the same size, you can compile them into that sort of big panel that I showed you. And you could have zooms on there or not, or you could have them inverted or not. So the idea is you make these sort of rows and then you stick them together with the Montage Compiler. That's the idea. Just want to show you one other trick with this Roi Zoom business. So maybe this spot was interesting to us, but perhaps we want to also focus on this spot here. So what we can do is we can actually add another zoom on top, and we can do this up to 4 times because we've got 4 corners of the panel to play with. So let's just do that, but we don't want to add another white box because then that would be a bit confusing. So what we can do is pick a different foreground color up here. So let's just pick blue for the sake of this demonstration. And then what we'll do is we'll do another Roi zoom. So this time we won't put it left bottom because we've already got one there. We'll leave it left top. They could be different sizes if you want, but let's just do the same because it'll look nice. And what we'll do is we'll uncheck this box. So white border just overwrites whatever your foreground color is. So if you uncheck that, it will use the foreground color, and we've selected cyan. So now when I click OK, we can just specify this. Oh, let's go for this guy here and click OK. And now we get another highlighted one, which is sort of color coded. So it helps the reader to quickly see what's going on and why these 2 spots might be interesting. OK, so now as long as you can remember which zoom, so because we've done this twice, the file's called Zooms, Montage, So as long as you can find the right files to put back together, you can assemble your final panel. So what I'll do is I'll open that one, and I'll open the inverted one we did as well. And let's stick those together. Now you wouldn't have this as a figure. I'm just showing you that it's possible to put whatever combination you want together. So let's just make sure we've got them different. Okay, we're going to include a scale bar. Let's remember it needs to be the right size, and we click OK. And this is it's slightly horrendous, but it shows you everything that can be done. So we've got a normal one here, an inverted one here, and then we've got one with lots of Roi Zooms here. Okay, so that's the figure maker. Now, just another couple of minutes, promise, won't take too much longer. What I did was compiled the montage there from top to bottom, and the merge was on the right hand side. Now it's possible you might not want to do things that way, so I'll just show you an example of doing it the other way around. So for example, in this figure here, if you look at B, you can see that the merge is on the bottom, and what we've got is time going this way. It makes more sense for time to go across the image, and therefore your channels should go down the image, whereas in these sorts of images it's better to have the experimental conditions going down and reading the figure across. It just helps with understanding what's going on in each of the panels. So how do we do this? We're now experts at all this, so I can go quickly through this, but I've got these already saved as distinct frames. So they've already been chopped, and what we do is we go to our make montage directory. So we select the files we want. If you remember, we have to specify where they're saved, so we'll go in the same place. We have to know something about the image, so that's what we'll do. 3 grayscale panels, 1 merge. Now we click this vertical montage thing, so that's what that was all about. Instead of stacking this across the image, it's going to stack them downwards vertically. If you don't check that, it doesn't know, and it will do it the way that the previous example was done. So again, we need to know what's what. I'm just going to put anything in here, just to say that you can do whatever merges you like here. It's just our lab preferences for RGB and a maximum of 3 channels shown, unless we really can't help it. Grout size is 8, but these are very small actually, so I'm going to change this to 4. We'll leave the scale bar for the moment, and it starts to make these, as you can see here. So we're getting our vertical montages now. So what we can do is then stack them together, and the way that works, you saw me do it before, but this is important. So Montage Compiler, we go again. Now it says here, detected columns, select order for your compilation left to right. I don't expect you to remember, but when we were doing it last time, this box said from top to bottom. So the code knows that we've got a vertical montage and we need to stack them from left to right. Okay. Now, if that's not the case and you wanted to do something different and stack them top to bottom anyway, even though they should go left to right, you can click this box here, force other orientation. And the same applies to if you've got rows and for some reason you want to stack them across, you can force the other orientation by ticking that box. But for most usage, you wouldn't need to use that. So that's what that checkbox is about. Anyway, we can just put in here the order that we want. So we're just doing them in time order. 6. If you remember, we changed the grout, so let's move that to 8. Let's put a scale bar in. Actually, this is the correct scaling for this image because it's from the ultra view, and we're going to do, I don't know, 2 microns because it's a very small image. We click OK, and we get our lovely montage here. It goes across the image now with these vertical montages going down, and the scale bars in the corner. It's a nice size. And again, we get an output here of what we did. So that force other orientation, you might want to do that if you wanted to put together, you know, two rows. Say you had two conditions that were going to be side by side in a big panel. You could put those two rows together one by one, and then compile the montages the other way. There's no limit to the number of times you can run the compile montage, so you can make really big montages if you want by sticking lots of montages together. It can get pretty wild. But anyway, I've just showed you a sort of basic example there of what the code can do and how it can sort of simplify and make figure making more reproducible. Just to say that there's a few other goodies in here. I'll just very quickly show you. So this example here, we can check for color blindness. I'm sensitive about this because a reviewer recently asked us to check our images for color blindness. So this is just a way that you can have a look at you know, the original images here, and this is what a colorblind person might see with your image. So you can check to see how accessible your images are with different types of color blindness using this little command here. So that was there. We've got some movie annotation tools that, well, they're kind of complicated, but there's description of how to use it on my GitHub page, or I could do another drop-in some other time. And we have some utilities that are perhaps not so widely useful in there. There's also a blind analysis thing that we use in the lab for taking the names off images and removing any identifying character, so that if any manual analysis needs to be done, you can do it blind, so you don't bias your analysis. And there's this Eln Saver, which is just a quick way to save a low, it's not low resolution, it's quite high resolution, but it's a low file size version to put in your electronic lab notebook if you're doing some kind of analysis and you just want to take a snapshot. Most people just take a snapshot on their computer, but it's a way of doing it in ImageJ. And that's it, really. That's the code. So